In a study recently published on the preprint server bioRxiv*, the researchers applied their previously developed precision-cut lung slice model to compare the pathogenicity of coronavirus 2 with severe acute initial event to study human respiratory syndrome. SARS-CoV-2 infects and replicates in the airways and subsequently causes lung damage. Unfortunately, in hospitalized patients, host interactions with SARS-CoV-2 have been found to persist for days or weeks. This makes deciphering the initial host response to SARS-CoV-2 a challenge. Although animal models benefit in this regard, they do not fully recapitulate the complexity of humans. About research. In this study, the researchers applied their previously designed PCLS model to mice to study the primary host SARS-CoV-2 interaction in humans. Aerate human lung lobes with 2% low melting point agarose and obtain PCLS. Sections were directly infected with SARS-CoV-2 for 3 days, followed by immunofluorescence and flow cytometry analysis. Cells were stained for double-stranded RNA and SARS-CoV-2 spike protein for further study. In addition, PCLS were infected with influenza A virus or SARS-CoV-2 to assess changes in lung composition and gene expression caused by SARS-CoV-2. To study the effect of SARS-CoV-2 on myeloid cells, an angiotensin-converting enzyme 2 expression screen was performed on alveolar macrophages residing in the air spaces and thus directly exposed to SARS-CoV-2 analyze. To study the ability of EMs exposed to SARS-CoV-2 to generate and release new viruses, bronchoalveolar lavage multiplicity of infection was performed on human lungs after incubation of SARS-CoV-2 and IAV with BAL cells. 0.1 or 1. Plaque assays were performed to determine viral load generated from BAL supernatants. A similar amount of virus was incubated with medium alone as a control. After 48 hours of incubation, cell-free supernatants were collected and used to infect Vero E6 cells with SARS-CoV-2 strain USA, WA1, 2020, or Delta strain, and MDCK cells with IAV. 24 hours after infection, cells were scored by flow cytometry and S staining. In addition, coronavirus disease 2019 from 7 intubations.